Okay, let's go ahead and get started. It's been a little bit of a hiatus for us here on the seminar series, but we're really excited to be kicking things off again. And so, for those who aren't familiar with iOS, so we're the Network for the Integration of Speciation Research, and we're a special topic network funded by ESEB. And the network's really brought together a bunch of people that are really interested in integrating the field of speciation research. We have a bunch of different goals, which include things like outlining best practices for data analysis and generation, all the way through to fostering diversity and inclusion in the field. And I think the seminar series has been a really important, part of helping people share their different perspectives on speciation. And so, really excited to kick this back off. If you're interested in, getting notifications about seminars, or you want to watch past seminars, or just learn more about the network, you can use this QR code to get to our website. And given that it's been a while, that we have had some new changes within the structure of the committee, and I think what's really exciting for us is that we have four new committee members, which who joined us through a kind of competitive recruitment process. So super excited to welcome Jonah, Parvati, Rowan, and Hilda all to the team. They brought huge amounts of energy, and a big part of that has been kind of re-establishing or revamping our seminar series to take on a new format. And so with that, I'm going to hand over to Hilda and Parvati, who are going to tell you more about the seminars over the coming weeks, and then also introduce today's speakers. Yeah, you're muted, Hilda.
I just wanted to very briefly tell you about what we have in store with the seminar series. So it was Rowan, Parvati, and I that kind of joined the network and got the task to kind of restart it and think of whether we wanted to make any changes to how the seminar is run. And we were helped by this by feedback that we got from the community through a survey that had been circulated, which indicated that people generally liked the format, but that they requested some more early career researchers being involved, more geographic diversity, and maybe some occasional format changes. So in response to that, we've come up with these three themes for the next year, where each theme has three sessions included in it. And the first theme that we'll hear from today is our early career speciation researchers theme. So very much kind of catering to that request from the community. And in this theme specifically, we thought it would be fun also to include a bit of a Q&A with some kind of established speciation researchers of the choice of the speakers to hear something about like what it's like building a career in speciation research, basically. So that's going to be a first time that we'll be doing that today. So I'm very excited. And then another new thing we're introducing with the seminar series is to have a workshop which we're planning for towards November, which will be more about getting some hands-on experience with methods that people use in speciation, and it's yet to be confirmed what exactly the methods will be and what that will involve, but we'll keep you posted. So if you sign up to the mailing list, you'll be well informed about all of that. So with that, I'd like to introduce our first speaker of today. I'll just stop sharing. So our first speaker is José Cerca. We're really grateful for him for giving a talk to us today. José did his PhD at the University of Oslo with a visit to Berkeley, and then came back to Norway for a postdoc at the Norwegian Center of Science and Technology. And now he is a SciLife lab group leader at the in Sweden, in Stockholm, still, and his work is very much about islands and archipelagos, and all the exciting evolutionary and ecological questions that they can help us answer. So I'm really intrigued by your title and looking forward to your talk, José. So please take it away.
Thank you. And thank you everyone for being here. Desktop number 2. It's no pressure at all when the whole field is here. So first off, thank you so much for having me, and it's really a pleasure to be here because when I was a master's student studying ecology, I always dreamed to study speciation, and now I'm making the argument that I don't study speciation. Me and Enrique, we have this paper submitted that I would say the title is quite provocative, but hopefully you will see our point of view and perspective in the end. I also want to say that, I'm really exhausted because my 2-year-old did not allow me to sleep, and I have taken a flight today. So I'll try to be coherent and I'll try to do my best. Um, Before I actually talk, I just wanted to, like, I haven't been to the other seminars, but I really want to say that this presentation is dedicated to David Marquez, who I met only on conferences like many of you, and it's hard not to get emotional because David passed away, and um, to me, David helped me a lot because I was struggling with a toxic PI at some point, and he took many hours of a conference just to sit with me and to give me advice, and so um, it's just really hard to see someone so good pass away. So yeah, sorry for starting with that tone, but I wanted to make a proper dedication to someone who I really admired and looked up to. So before I start, I wanted just to thank the people who have contributed. And I mean, me and Rika, we are publishing this work, but a lot of my friends and people who are here have read this, including Mark Ravine, who's talking later, Jasper Bowman, Kyle Lusek, Lorena, Ament Velasquez, Alex McIntosh, Marius Roesti, Alan Lemon, Ken, and two anonymous reviewers. And thank you so much for your support because um, this is not an easy topic, and you guys really helped us make the most out of it. So studying speciation is hard, and I'm preaching to the priests here, right? We all know that there are a lot of gray areas, so things do not just there's not easy tipping points. I particularly like this slide here on the left, this image where reminds me of kids saying, "Are we species yet? Are we species yet? Are we species yet?" I think it represents well this type of issues and where to draw the lines. One thing that became very clear from the last few years, and I really like this paper from Stankowski and Ravine from 5 years ago now, that they give this overview that speciation is a continuum, which is not a new idea, but they brought it into a really nice framework. And this means that there are no clear tipping points. Populations might be in different parts. So, for example, if you have a species with several populations, the pairwises might fall on different ends of the continuum. There might be reversals. There might be things, and this makes also this adds a layer of complexity to speciation. There's one funny thing about speciation research is that we cannot agree on a common language. And I really like this paper by Richard Harrison, the one on top here on evolution, that just goes on about and says, like we use the same terms in different ways and in different contexts, they can mean different things. And there's an argument to be made that this does stop a bit the progress in the field. And some things, like the integration has talked about this, the integration paper, but then I also highlight this paper from Westrom and Stankowski et al., and then the response from Roger Butlin, that they do touch a lot on this concept of language. You can also choose your favorite flavor of speciation. Textbooks will be populated with allopatric and sympatric speciation, but also other nice but complex things also happen, like these ideas of mutation order versus ecological speciation. And if you're not familiar, there's this paper by Wolf Schluter in Science that puts it really well. And then a really fun paper that I read recently by Maddie James that really goes on and shows that these are not mutually exclusive. They are just, ends of a continuum, but they can happen in the same adaptive radiation. So, and in the same set of lineages. So studying speciation is hard, but also because of this. And one paper that I really like from recently is this paper from Resti et al. That came on the Cold Spring Harbor collection that basically just says, every rule in speciation research has an exception, and this makes things hard, meaning that we cannot predict when speciation is going to happen or how it happens. And there's an argument to be made that studying speciation is fun because it's also hard. And again, just so everyone is on the same page, reproductive isolation is the mechanism of speciation. And when we measure reproductive isolation, we are studying speciation. And then coming back to this insightful paper from Anya and Sean et al., And one important distinction that they made, and I think it's made quite clear and nice in the paper, on both on their paper and then on the response by Mallet and Mulet, or Mulet and Mallet, is that there's this distinction between the organismal and the genetic measurements of reproductive isolation. And so just to give you an overview, organismal aspects include what's up here. That's the fitness measurements, the hybrid crosses, assessment of assortative mating, but then things like genetics might be genetic differentiation, a divergence, migration across the genome. And they have this really nice representation here that you might think, that you might see, there's these birds and the camouflage and the song might lead to this accumulation of reproductive isolation. But when we think about genetics, we would think about in these terms of, migration, effective migration, etc. However, one thing that I noticed in the literature as I've been following now speciation is that there seemed to be some degree of disagreement between both. And I came across this paper. Lorena made me aware of this paper of hers from her and from the lab of Hanna Johansson here in Stockholm, where they take these genetic matrices, and if you're used to studying genomics like I am, these are extremely low numbers of FST, genetic differentiation, and the XY, which is genetic divergence. But then when you look at the organismal metrics, they write something like prezygotic barriers prevent more than 95% of the fruiting body production in some crosses, but then there are additional postzygotic barriers acting subsequently. So this means to me that there's just some degree of discrepancies between these type of measurements. And so the working question that me and Rike were trying to solve is whether there are these discrepancies between the organismal and the genetic aspects of reproductive isolation. And so the first thing that we got interested was we did a literature survey to try to figure out, because we wrote the perspective, not a review, whether there was a balance between organismal and genetic metrics. And we focused on the last 5 years because, genomics is recent, and then there's the bias from COVID that was quite clear from the data. And analyzing 100 papers from the last 5 years, I divided this into one data type and multiple data types. And what is really sticking out from here is that a lot of people are using genomics. Genomics is used, 60% of the works that we analyzed only included genomic data, 10% only phenotypic data, and 2% experimental or process data. But then when we start adding up, for example, these multi-layer data, for example, you see genomics here, 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 and here, and here, basically 85 to 86%, I think, of the works have included this type of data, which then suggests that in terms of data structures, there seems to be a very large imbalance in terms of genomics. And then the second question that we were interested is, okay, given that these works rely so much on genomic data, is there a conceptual balance in the way that they look at this reproductive isolation? And so basically here on different colors, you will have the measurement of reproductive isolation, whether it is genetic and organismal, and then you just have the number of studies as a lollipop plot. And you will see that the blue ones here that really stick out with more than 20 something works, and it doesn't add up to 100 because not all papers would include this, but the ones in blue are the ones that really stand out, are the ones that are genetically oriented. So people measure genetic differentiation, genetic divergence, gene flow or hybridization, and they would conclude that, for example, the cessation of gene flow would be an indication of reproductive isolation. So there seems to be a large imbalance here too, which is not surprising because if people are using data structures from genomics, they will also use same concepts. But then we were interested in, in the papers that were a bit older and the papers that were trying to look at some degree of organismal reproductive isolation and trying to look at how is this correlated with some degree of genetic differentiation, genetic divergence, etc. And so you have seen possibly in different works, and for example, Koinen, some plots that look like this. You have the genetic divergence here that goes from 0 to 1, and reproductive isolation that goes from 0 to 1 here. And then the question is focusing on this axis, whether we can find a correlation. And we came up with this, let's say, prediction or this model or this just way of just slicing up the data where we're like, If it's quartile 1, which is the gray, this means that reproductive isolation is high, but there's low genetic divergence. Quartile 4 would be that you have low reproductive isolation with high genetic divergence. And then what we did is, and then we would say if we would expect this to be associated and correlated well, we would expect things to be falling mostly on these two quartiles, which is in the red colors. So then what the data shows here is that when we analyze the papers here, and sorry that it's a low quality that you cannot read the papers possibly, what we see is that in a lot of the cases, a lot of the data seems to be falling on quartile 1 and 2, which would be the ones that, let's say, do not draw this association with the correlation. Of course, this analysis has some issues if things fall closer to where the axis, the horizontal and vertical lines fall, but like you do see this from several data sets. And then the fourth question that we were interested was focusing on genetic differentiation and divergence if there are many outliers on 95% of reproductive isolation in early divergence. And so going back to this one here, we were basically interested in this point here that I'm putting here. So for example, you will see that this point is around 1 to 99 or something, and this will be the earliest in the data set that we will find divergence. So for example, the divergence here would go from 0 to 1, and then at the value of 0.02, more or less, we would find one pairwise comparison where there would be reproductive isolation. And what we find out here is that a lot of these taxa seem to have a lot of these dislocated points. So the way that you read this is that the different colors on the left are different taxa with plants, amphibians, etc. And I want you to know, and this is clearly, I think, a weakness of this analysis, but it's hard to make this because a weakness is that we have allozymes, we have rat-seq data, we have whole genome data here. It's a mix of everything. But then what stands out and what I want to highlight is that we have the yellow color and the blue color. The blue color is the maximum level of divergence on the data set, and the yellow color is the divergence value of that point that I showed you that has more than 95% of the data. So for example, in this case, it means that the maximum 95% is just percent of reproductive isolation is achieved at the level that is very close to the overall divergence of the data set, whereas a case like this, it means that you find a divergence, like the biggest pairwise comparison between divergence is 1.17, but you already find reproductive isolation of 95% on individuals that have, oh, now I have written over it, but 0.02. And so basically this is showing that there seems to be some disparity between the overall levels of reproductive isolation and divergence, also with where reproductive isolation actually becomes. And so part of what we say that is this identity crisis, so to say, is how do we put this into the perspective of what we have learned over the last few decades of speciation research? And one of the questions or one of the things that we discuss and focusing especially on these two big lollipops in blue is whether the cessation of gene flow is a good metric of reproductive isolation. And sorry, this slide is a bit wrong. There's a really good discussion on the literature for this, and like I had totally missed this out. It was one of the reviewers that pointed this out. And basically, Sean and Anya wrote a paper, the co-first authors, suggesting that we should use this gene flow because it's quantifiable, it's a good metric, etc. But then there are some really insightful comments from Leonid Moyle, Roger Butlin, and James Mallet, and Sean Mullet. And basically what they say, which we agree, is that perhaps not. Perhaps gene flow might not be the best indicator of reproductive isolation, but then of course it is convenient, it's quantifiable, etc. These arguments are part of Stankowski and Westham. But then what these three papers focuses is that it's not integrative of organismal traits in a field that is pluralistic. It conflates geographical isolation and demographic history, also with demographic barriers. And so for the genetic differentiation and divergence, should we consider it as a good metric of reproductive isolation or not? And here, I think I would like to make a distinction. And for me, almost reading the journal, that the paper, after having scored the literature, towards the end, I could see what type of metric was going to be used for reproductive isolation almost by the journal, because of course, like more phylogenetics and systematics journals, we'll use just branch length differences, etc. And the argument that we make is that perhaps not very broad measurements of FST and DXY can be good indicators of reproductive isolation, but I think that there start being many good examples of genomics, and I'm highlighting the work of the people who have read. So, for example, in this paper, Alex McIntosh, who's my postdoc now, what they did was that they studied how these breakpoints of structural variants and these breakpoints of fusions and fissions will have their migration on those parts of the genome and try to figure out the association with recombination and other aspects with a part of the genome. And then this paper from Jesper, where they did crosses, but then did the genomes of these to try to figure out what was the association between the inversions and the associations between genomic divergence and etc. with this. So I think like we're going in a good direction. It's just like perhaps these very broad FST and DXY measurements might not be ideal. And then whether to the field is overly dominated as genomic data, I speak against myself because I have done my career as a genomicist and someone who lives and breathes genomic data. But this I leave you to decide and interpret because I think it's quite clear from our data. But then, of course, I would like to be a devil's advocate here because, of course, if we are going to do only organismal measurements of reproductive isolation, it's not feasible for some systems. They are hard to get. Some systems we cannot bring to the lab. I mean, I studied trees from the Galapagos. It's impossible for me to bring them in the lab and ask some of the questions that a lot of you try to answer. Um, and then, of course, genomic data is more convenient. And then there's this aspect that I haven't stressed, but it comes from this discussion I showed you from Stankowski, West Ham, and the associated commentaries, that genomic data does give you an insight on longer term perspectives, whereas these crosses sometimes can be just something that is happening now, but it might not be reflective of the evolutionary history of the lineages. So the way forward is to be integrative, of course, integration of speciation seminar series, and to test hypotheses. I think like something that hasn't been tested, but now with more reference genomes, is looking at chromosomal speciation. And I'm thinking, for example, on a really seminal paper by Lauren Riesberg from 2 decades ago, where he lays out a lot of hypotheses on chromosomal speciation, and also trying to rethink the dichotomy between intrinsic and extrinsic barriers, and try to figure out how do we better conciliate the fact that these things might not be correlated or associated. Um, Otherwise, and one thing that we conclude is that if you're not familiar, the law of the instrument is when your tool is a hammer, you will see all your problems as a nail. So you're just going to be using the hammer. And this is a cognitive bias, meaning that when you have one tool that you know very well, you just use it a lot. So thank you for me, tack för mig. Thank you for having me in Swedish. And this nice photo is from my friend Gonzalo Rivas Torres with our system in the Galapagos. And these guys really love these these glaciers really love this middle of nowhere where they can live alone on these barren lava rocks in the middle of the Galapagos. This is one of the most recent islands, which is now under volcanic activity and eruption. So yeah, thank you. I don't know how the format is, but I think I will happily try to answer questions because yeah, I'm really exhausted, but I'll try to do my best.
Thank you so much, Jose. Yeah, certainly we can have time for some questions now, specifically about your talk, and then just at the end we'll have more general discussion in this Q&A with Mark. But right now, yeah, please, if you have a question, you can either just unmute yourself and ask it yourself, or raise your hand or write in the chat, and I can read it out for if you feel more comfortable with that. So I see 2 hands raised. Maybe Sean, do you want to go first? Because I can see your name. I have to figure out who the other's
Oh,
hand is.
yeah, thanks so much, Jose. That was really interesting. Um, yeah, so I guess I got some questions and some reactions. I think the first thing that I guess I would highlight is that there is this big difference in time scale with what these things are measuring. So, like the estimate of a barrier in one cross, if you think about like a measure of RI of 0.95, that seems really high, but actually over evolutionary time frames, it's not really high at all, actually. So it's actually quite low. So actually, we wouldn't necessarily expect that to have a really marked effect on genome-wide differentiation. And I guess maybe the other thing, another comment would be that the measures of the genetic measures that you've used here were certainly ones that Westrom et al. Suggested that aren't necessarily good indicators of gene flow anyway. So favoring things more like demographic inference. So I think it would be really interesting to see, like using the methods like that Camille Roux developed, whether you see something similar. And then I guess a final question for you is if we're not measuring the organismal barriers to understand patterns of gene flow, why are we measuring them?
Um, well, to start with the clarifications, I mean, we tried to do justice with that, and I mean, at the end of the day, I think we wrote what hopefully is a provocative paper, and hopefully what just puts us all thinking. It's not to criticize because like I don't agree with Westham and Stankowski at all, but like I think it is a nice paper in many aspects, and it generated a really nice discussion. And as you said, like I alluded to this only briefly, but yeah, the timescales are really an important axis that we have to take into consideration here. Um, your question was, if I understood well, why are we not measuring gene flow if we are studying reproductive isolation in the organismal aspects, right?
Well, no, it was that, the reason that people are interested in measuring RI at the organismal level is that they think it tells us about the discontinuity of gene pools. So if it's not affecting gene flow, and we don't think it's a good measure of that, why would we study that at all?
I mean, it's just like it's just these levels of complexity. It's not very it's very hard for me to answer that on the spot here. I think gene flow, as you guys have argued, gives you like this very quantifiable and this very comparable metric, but and that focuses on these long time scales. But I think one thing that we want to highlight with the paper is not saying this is not important. It's just saying there is a clear imbalance now on the field with terms of the data, we think, and perhaps that needs to be corrected or not. I mean, that's up for the field to decide. And I mean, hopefully, we make points that the next people that are sitting in grant committees think, oh, wow, cool, a more experimental type of work instead of a much more genomics type of experiment. We need more of this. So I would argue that both perspectives are interesting, and of course, one is going to reflect the other because if organismal stuff is not, if you can't cross, then there won't be some gene flows. That there's going to be a degree of association. I'm sorry, I don't think I gave you a very clear-cut answer, but
That's great. Thanks so much. Cheers.
yeah.
Joanna, do you want to go next?
Yeah, I actually had very similar points to what John already said. But first, thanks so much for a wonderful talk, Jose. Yeah, I also think like the time aspect is really important. And so some modeling approach to figure out the extent of gene flow in addition with the time scale, because you can have super strong reproductive isolation, but very recent, and then you would still have very little genomic differentiation, or you could have allopatry without any reproductive isolation, and then you could have lots of genomic differentiation. And so the geographic aspect of it also needs to be considered. And so I think that genomic differentiation kind of has the time scale and geography plus reproductive isolation, and reproductive isolation not just at the specific trait, because usually people measure how much does this trait contribute to reproductive isolation, but then the gene flow is kind of across all different reproductive isolation barriers. And I was just wondering if you had any more thoughts about kind of like the total reproductive isolation versus because we can't measure all the possible contributors. We think that specific contributors are important and we measure those, and that is very important. But if we find low reproductive isolation from that contributor and gene flow is very low, that means that there's others that we haven't tested yet, and how to go about that.
Could you repeat your question? Sorry.
So because the gene flow is, as I said, the time and the geography is important as well. But in addition, one of the problems for measuring reproductive isolation with like tests of, for instance, assortative mating, then we only test one component of reproductive isolation, right? Yeah. And it's kind of the accumulation of all the different components that really affects gene flow.
Yeah.
And so we have certain suspicions which traits or components might be important, but we can never measure all of them. What do you think about that? Like
Yeah, I don't know. I think it's quite system specific and it's quite also question specific, I would say. And I mean that is the magic of genomic data and also why I like it so much, that you can just go and do genomics and you get so many nice answers and so much more than the answers is it just gives you this overall evolutionary history. So I think, I think both my and Rika's, as I have answered through Sean, because I guess your question is a bit similar. We just want to bring this question: is the scale very tilted or not? Because we're not saying genomic data is not important. Both me and Rika are genomics people. I just think that then for the speciation research, how can we better accommodate both? And sorry, I understand this is not a clear-cut question, but I mean, I guess this is the point of the seminar, is in being integrative and perhaps, other people have better answers than me. So sorry for that.
No, I think that's great. And like making people think about it is very important and very nice. Thank you.
Yeah, Jim, Mala, do you want to ask your question?
Yeah, there's various chats here asking you to share your preprint, which I think everyone would be interested in. Jose.
Oh, thank you. We don't have it as a preprint, and I don't know why, but I will share a version. We're just now working for the revision, so I will share perhaps next week. Perhaps I will take contact with Hilda. But thank you for the interest.
Any more questions for Jose?
Jim, do you have a question?
Yeah, I could do. If nobody else does. Maybe neither gene flow nor reproductive isolation is what we need to measure if we're going to study speciation. If we want to define species, perhaps it'd be better just to look at multiple locus differentiation in sympatry.
Oof. That's hard for me to answer. But yeah, it's just then.
I mean, obviously, reproductive isolation is what we mean that causes this differentiation in sympatry, right? And it's stable. But you could have high gene flow and maybe high levels of selection against that gene flow, or low gene flow and low levels of selection against that gene flow, and you'd reach the same balance. So I think that's the problem with trying to use reproductive isolation rather than using selection, sterility, actual gene flow, I mean, I've argued this until I'm blue in the face and nobody seems to pay any attention, but it seems to me that's what's important. I mean, we realize now that lots of species hybridize, and yet we can call them separate species. We can tell them apart. So what do you think?
Um, I mean, we could go back to those classical problems of like we have to find things in symmetry, right? When a lot of things are in allopatry. But again, I would have to think about that. And sorry for the really boring answer. I think this
But
is why.
you mean, I think, even when you've got allopatric species, I think you mean to say that they would be stable if they were brought into sympatry. You don't want to just name, geographic variants,
Yeah, but
right?
then
So
it's not
as a
easy to
null
do that.
hypothesis, they're the same species, unless you really think that they wouldn't blend completely in sympatry.
Yeah,
I,
that
th
makes
I
sense.
think, yeah, I think Joanna was mentioning this, like it's the geography matters. And so, reproductive isolation is something that's independent of geography. And uh, in the sense of a reproductively isolated species, you don't mean that it's always defending against gene flow. You mean that it would if it had to. I think, right? Maybe I'm going crazy. Just tell me I'm crazy. That's fine.
You're not crazy, Jim, but I think we should move on to the next speaker, right?
Yes,
So we don't
please.
keep everyone.
Yes.
So yeah. Okay,
Yeah,
great.
thanks, Jose, again for a thought-provoking talk. It's great to start with this energy. So for our second talk today, I'm very excited to introduce Andrea Estandia. She completed her PhD at the University of Oxford in Sonia Clegg's group, and she is currently a postdoc also at the University of Oxford with Ben Sheldon. She has worked on several questions that looked at process underlying adaptation and speciation using genomics. And today she'll talk about the consequences of colonization in the silver eye. So on to you, Andrea.
Thank you. I'm gonna try to share my screen. Can you all see that? Yeah. Okay. Yeah, hi everyone. My name is Andrea, and thank you very much, first of all, to the organizers for inviting me to give this seminar. It's an honor. And yeah, first I would like to introduce you to my institute, the institute where I work at the University of Oxford, which is the Edward Gray Institute of Field Ornithology. And this is the latest photo we've taken a couple of weeks ago. Most of the people you can see here are PhD students and postdocs, but among all these people, there are also 5 PIs that lead 5 research groups looking at many different things, from speciation to several questions about ecology and evolution and how climate change affects ecoevolutionary dynamics, to biomechanics, etc. But I did my PhD in Sonia Clegg's lab looking at silver eyes and speciation of island birds. She works on the South Pacific mainly also on silver eyes, but also on other species like honey eaters. And she manages the long-term field project on Heron Island, which is an island off the coast of Queensland. And she focuses on questions about natural selection, demography, life history traits, things like avian malaria, etc. And now I'm doing a postdoc with Ben Sheldon, also at the same institute. And he also works on a long-term field project, this time in Whiten Woods. And this project has been going on since 1947, so a very long time. And here we mainly monitor gray tits, also blue tits, and we have also incorporated winter moths and oaks. So we have the whole trophic network. So we can look at, for example, how climate change has affected some of the levels of the trophic network and how that impacts the rest. And the field project basically focuses on questions around natural selection, life history traits, and social ecology. But today I'm not going to talk about my postdoc. I'm going to talk about silver eyes and evolution on islands. So we see that there is repeated evolution all across the tree of life. We see repeated evolution of freshwater, of the evolution towards freshwater ecosystems in sticklebacks, the evolution of ecomorphs in Hawaiian spiders, the loss of flight in rails and many other birds. and dewlaps in anoles. And one thing that all of these animals have in common is that they live either on lakes or on islands. That doesn't need to mean that there's not parallel evolution on the continent, but islands are somehow special because they impose similar selective pressures, and that can drive repeated evolution through natural selection. And one of the evolutionary consequences of island colonization, one of those predictable changes, is what we call the island rule, which is the repeated evolution towards intermediate body sizes. It is a rule that applies to many animals and some plants as well, but today I'm talking about birds. In birds, we see an increase in body size, and this is a consequence of ecological release. So on islands, we find that there are fewer predators, and then birds can increase in body size. And then we have the speciation process on islands, and I like to use the taxon cycle proposed by E.O. Wilson in the 60s, and when he was in Northern Melanesia studying ants, and he hypothesized that speciation happened through different stages of range expansion and contraction. So the first stage could be an expansion in which the species has high dispersal ability and colonizes all the islands or many islands across an archipelago. And this is what in the bird literature we refer to as the supertramps. Then a second stage in which those populations lose the ability to disperse, and that leads to population differentiation. And that is what in the bird literature we call great speciators. And this is the stage in which I'm going to focus for this talk. Then we see extinctions on small islands due to small population sizes and stochasticity, and the completion of speciation on big islands. And then finally, we find an altitude specialization and more extinctions. So, great speciators. Great speciators were basically those birds that Mayer and Diamond found in the Northern Melanesian region, where E.O. Wilson proposed this theory in the 70s and 60s. And basically they found that there were some birds that followed this pattern of differentiation. And they compiled a list of 31 species. And this is not phylogenetically constrained, so we find members of the kingfishers, of doves, parrots, basically all across the tree of birds. But probably the most famous great speciator is the genus Zostrops. So within Zostrops, we find many species that show this pattern of subspeciation. And this is a genus that has more than 120 species, and it is the second fastest diversifying lineage with invertebrates after cyclids. And they mostly live on islands, so we think islands have to do a lot with this rapid diversification. And one very interesting species within the genus is the silvereye. The silvereye has 16 subspecies with a wide continental, maybe in Australia, and island distribution. They have a generalist diet. They feed on insects, fruit, nectar, seeds, and that is key to their success as colonizers because they can arrive to an island and basically just eat whatever. They live a relatively long life, and even though they have small clutches between 2 and 4 eggs compared to other passerines, they recover really well from bottlenecks. They're highly social. They usually migrate or disperse with family, friends, neighbors, and they pair for life. And they do this very cute thing called allopreening. And in some places, they're considered a pest because they love fruit. They also attack or destroy orchards in places like New Zealand. So this is our protagonist for today. And we are going to answer some research questions. First of all, we need to understand the evolutionary history of the silvereye in order to answer the rest of the questions. So we're going to do that first. Then I'm going to try to answer whether water barriers drive diversification better than continental distances or continental barriers like deserts that in Australia can, have a big impact in the separation of different populations. And then we're going to look into what are the genetic mechanisms that underlie those predictable shifts in body size on islands. Basically looking at the genetic basis of the island rule. So the silver eye, what do we know about it? We know that it originates somewhere on the Australian mainland. And there are different waves of colonization to different archipelagos. The first one, we think, happened to Southern Melanesia. So these two archipelagos, New Caledonia and Vanuatu. Then a second colonization to Lohau Island a bit later. Then a third one to Heron Island, which is very close to the mainland, but still we find a different subspecies. And then from Tasmania, which is very well connected to the mainland through migration, we find a sequential colonization to the South Island of New Zealand, to Chatham Island, then the North Island of New Zealand, and then finally Norfolk Island. And this last colonization is very recent, and we have historical records for it. We also find a human-introduced population from the North Island of New Zealand to French Polynesia, which happened like very recently as well. And in French Polynesia, they keep colonizing new islands over and over, even nowadays. So this is what we thought the Silver Eye evolutionary history was, but it was based on cytochrome B3. So I wanted to revisit this for my PhD. So I used samples that had been collected over decades because look at the spread of these samples. They are all over, and it is very difficult to work with vertebrates in all of these countries, very difficult to get permits and to get blood samples. So yeah, it took a few decades to get all of these and many people involved. And yeah, I used 114 individuals spanning 8 species within the genus for mainly for outgroups, and then a few subspecies within the silver eye. We generated whole genome sequencing at 5x, so semi-low coverage, and we got 22K high-quality SNPs to do phylogenetics with it. And I'm not going to show you all the phylogenetic trees that we have in the paper and all that because it's very tedious. But just to say that basically we find like 3 different clades. So first, the other Zostrop species separate, which makes a lot of sense. And then within the silver eyes, we find that Southern Melanesian is a bit more separated. And this also makes sense because is what we thought it was the older colonization. And here again, like not looking at the details, just to say that in general, mainland populations and recent colonizations do not show as much structure as islands do. So islands in general show quite a lot of structure, except for this part here in central Vanuatu, which is very well connected. Well, not that well connected, but a bit better than the rest of Southern Melanesian populations or Heron Island and Nohau Island. So that made us question whether water barriers led to higher population differentiation. So we built a model in which we tried to predict pairwise FST with geographic distance between populations and whether the comparison was island-island or continent-continent. And we added a multi-membership grouping term because we are including the same island in multiple comparisons. And the results speak for themselves. We see that island comparisons actually have higher FST, and that FST is kind of increases with distance very sharply. So even if islands are really nearby, we see that populations have higher FST than on the continent. And bear in mind that all the samples across the continent in Australia are really far away, and some of them are separated by huge deserts. So is this how silver eyes likely evolved? Yes, basically our conclusion after building all the phylogenetic trees, yes, we're just shifting a bit the ages, the colonization ages to a bit older using a time calibrated tree, but mainly the cytochrome B3 agrees with our data. So we relatively resolved the evolutionary history of the silver eye, which is useful for the rest of the questions. And then, yes, we show that water barriers drive diversification more than continental distances and barriers. Cool, so we're gonna look into the island rule now. We see that the silver eyes tend to evolve towards larger body sizes on islands. They mainly originate from the mainland, and then the rest of the populations are sorted by colonization time. So the pink ones are the recently colonized populations, so they don't show that much of an increase. But then Heron Island, Lord Howe Island, and then Southern Melanesia, we see like that increase in body size. So the mechanisms that are involved in parallel evolution that we can expect are a new mutation that can happen randomly, and then if it's beneficial, it can increase in frequency with time. Then we can also have standing genetic variation that is reused and also increases in frequency in time. This is mainly the main mechanism that we find plays a role in parallel evolution, but also adaptive integration. So when two related lineages come into contact and there is a beneficial allele that increase in frequency. So we sequenced 380 whole genomes from 31 populations and 13 subspecies of the silveri. We used probability-based bioinformatics, which is another way of saying angst, which I'm sure many people are familiar with. And we did a genome-wide association study with 10 million SNPs to try to find first the genetic basis of body size, and then just to try to find whether the associated SNPs were new mutations, standard genetic variation, or intercrest. And yeah, there are a few measurements that we use to calculate body mass. Here I have a little list. It's a shame that I don't have time to talk about fieldwork because it's the best part of the job, but hey ho. And yeah, these are the results for the Manhattan plot for the GWAS that we find that has our heritability of 0.6, which is a bit lower, but very close to what we find in the field for traits like tarsus length. So we have this population on Heron Island, and we have multiple years of collection, so we can estimate heritability from the field. And tarsus length is 0.7, so this is very, very close. And yeah, basically we find that there is an association peak on chromosome 2, and we're gonna look into it a bit more. First of all, just to say that these alleles seem to be private to the oldest colonization, so Southern Melanesia. Here I'm showing Central Vanuatu because it has not much population structure, and even within these populations, we find that those individuals with the B allele actually show an increase in the trait. And if you look, you do a gene tree of the region, you find that there is not much variation, and there is a lot of variation within Vanuatu and New Caledonia in comparison to another loci that or region that is associated with body size that is shared across all the populations. So this could represent standing genetic variation. And then if we do like some genome scans on the region, we see that there's an elevated FST when we compare it to the source, to the mainland, which makes sense because this is a private region. So it could be silly if it could happen. Then a reduction in pi, negative TGMSD, and a bit of a positive mu when we scan for a positive selection. So all of these leads us to think that it could be a new mutation. But it could also be other things, like for example, could be intergressed, and it could look like very similar. So Abby Williams, who is working with me at the moment, did like this beautiful scan and found that there is like actually no integration in the region. So this could represent the position of the associating SNP within the chromosome 2. So there is not an elevated FT. So just to wrap up, yeah, we think that a new mutation could play a role in the evolution, repeated evolution of body size, but in general, standing genetic variation actually is more present on the rest of the loci. So what's next? We have two projects around silverize that I'm still involved in, even though I've moved on a tiny bit from the project. One of them is the genomics of partial migration in silverize, and another one is hybridization across zoostrops. So for the genomics of partial migration, I did fieldwork in Australia sampling population in Tasmania and the mainland. So in this population in Tasmania, some individuals migrate to the mainland and some individuals stay. So I sample the migrants on the Australian mainland and the residents in Tasmania in a winter. And you can see that they're like super different, the birds from Tasmania and from the mainland. So the upper picture would represent the Tasmanian subspecies, and the one down here could represent the migrant. So they can be told apart quite well. So we can differentiate them when they migrate to mainland. So there's a PhD student who is currently working on all these samples. Well, currently, she's going to start in October to work on all these samples. These are a few pictures of our trip to Australia, which was very fun. And all the samples that we got, including fecal samples, to do a study on parasite dispersal as well. And anyway, the second project that we still have going on is led by Abby Williams, PhD student supervised by Sonia and by me, and in collaboration with Kristenberg. And we have gotten 16 Zostrop stopads from the American Museum of Natural History, including 3 hybrids. So we know, well, we think there is hybridization. There has been hybridization in the last century. So we got samples pre-hybridization, the 3 hybrids, and then post-hybridization at different points in time until the present. So we can explore things about the temporal dynamics of hybridization and things like that. And yeah, yes, I wanted to thank everyone who's been involved in all of these projects over the years. And thank you all for inviting me here.
Thanks, Andrea. That was a really nice talk. We have time for some questions. Mark has a question. Do you want to go?
Yeah, hi. Thanks for the nice talk there, Andrea. That was really cool. I just wondered actually, are there any other studies from other bird systems on the similar regions that show similar patterns?
About what? You mean like the evolution of body size, for example?
Yeah. The body size change. Maybe it was the migration as well.
So I haven't actually seen anyone working on body size that has found a new mutation, for example, involved in that. But if you have, just let me know because I could be really interested.
Well, yeah, okay. I don't know any. I was just asking out of interest. But I mean, are there any good studies of the genomics of body size change in island birds in any other systems?
I'm sure there are. Right now, my brain has gone like blank.
Okay, that's
But
fine.
yeah, sorry. Yeah, and in terms of like migration, there's a lot of from
Mhm.
Miriam Lidbogel's lab in Germany. She studies black caps and many other like robins, many birds that migrate, and looking at the genomics and way more. And Kira Delmore as well. She's looking into the mechanisms of migration really in depth. So it's a very cool.
But is my loss of migration a kind of common thing in island birds, do you think?
Yeah,
Yeah.
yeah. It tends to happen for sure.
So those studies aren't in, I mean, those studies aren't in island birds as far as I'm aware, right?
No, no. Well, Miriam has a study on robins in that I think loose migration on the Canary Islands. I'm not completely sure, but I
Okay,
think yeah.
cool. Thank you.
Maybe I have a question. Maybe this wasn't clear for me. So when you looked at FST, between like how it is differentiated within islands and in the mainland, was this the when you showed isolation by distance kind of plot, was this between island and the continent, or is there also a structure between the different islands?
Yeah, so there You mean like the plot from like the water barriers,
Yeah,
whether
that,
yeah. Yeah, so those comparisons were between continent and island comparisons.
uh,
So yeah, I mean, I think we also did a model in which we compare like island and continent comparison, but I'm not sure why we didn't include it. But yeah.
so I actually, yeah, got that the opposite. Okay, yeah, that's interesting. Thank you. We have time for maybe one or two more questions, if
I could ask one. So is FST the right measure of differentiation? Because I imagine island birds are bottlenecked and therefore maybe have low levels of variation. And levels of variation really matter in FST comparisons. In other words, you'll get a higher FST for a given DXY if you use if you have a low effective population size or low variability within a population.
Yeah, we also used DXY. It actually looks very, very similar. So, I mean, even though like these populations have suffered bottlenecks, in general, they recover like really rapidly. And yeah, I mean, like for the older colonizations, they're doing very well. For the new colonizations, you can actually see that there's a huge bottleneck and you can detect it very well. But for the other ones, I think FST is fine. But we also did DXY and it looks very similar.
we have a comment in the chat regarding there's a very recent major colonization of Southern California by Zoetrope simplex.
That sounds very cool. Yeah. I wish I had funding to study all those things.
Jordi, do you want to ask the question?
Yes. I was wondering, since one of the main question on this cool model that is Zosterops is that these birds are great colonizer or have been great colonizers and are great speciators on islands, whether um, they are people working on the dispersal ability and the migration. Coming back a little bit on the previous comments, but and what could be um, the genomic evolution of or the genomic background that allows for island birds to lose the capacity of migration or to dispersal, and that makes these speciation changes repeatedly across many islands. I don't know if my question is really clear.
Yeah, that's
Uh,
a very interesting question. So I have a paper where I looked at yes, the loss of dispersal across all of these colonizations. And I found that there were like some genes that shifted repeatedly after island colonization. And that didn't coincide with the neutral genetic structure. So there must be something genetic going on there that is related to some kind of personality traits or things like that, or more in the dispersal side of things. And then migration is another story. In general, it is the genes that underlie migration are a different set, and generally related to circadian rhythms and things like that. And in that same paper, I also find that there are differences between the partial migrants and the ones that migrate and the ones that stay. That's why we decided to do whole genome sequencing on the migrants and the residents to try to see whether there were like some differences. But those are not kind of those are kind of like connected. It is between Tasmania and the mainland, so they're not kind of involved in colonization of other islands. Well, they are a bit, because we think that the most recent colonization that arrived to New Zealand, it was a flock that was migrating north from Tasmania, and then due to a storm, they arrived to New Zealand. So they do have like some kind of capacity of dispersing, but it was just, I think, more accidental.
Thank you so much, Andrea. I think we should wrap up the questions there and move on to the last bit of today's session. So I'll just stop the recording first before I forget.